Now let's learn about fibronectin. The term matrix implies a structure made up of a network of interacting components. The term has proven very apt for the extracellular matrix, which contains a number of proteins in addition to collagen and proteoglycans that interact with one another in a highly specific way. Fibronectin, like several other proteins to be discussed in this chapter, consists of a linear array of distinct building blocks that gives each polypeptide a modular construction. Each fibronectin polypeptide is constructed from a sequence of approximately 30 independently folding FN modules, two of which are depicted. While FN type modules were first discovered in fibronectin, they are found as a part of many other proteins, ranging from blood clotting factors to membrane receptors. The presence of shared segments among diverse proteins strongly suggests that many present-day genes have arisen during evolution by the fusion of parts of separate ancestral genes. In fibronectin, the 30 or so structural modules combine to form 5 or 6 larger functional domains, illustrated by the colored cylinders in the figure. Each of these two polypeptide chains that make up a fibronectin molecule contain binding sites for the numerous components of ECM, such as collagens, proteoglycans, and other fibronectin molecules. These binding sites facilitate interactions that link these diverse molecules into a stable interconnected network. Fibronectin 30 or so structural modules combine to form 5 or 6 larger functional domains, illustrated, and they are binding sites for numerous components of the extracellular matrix such as collagens, proteoglycans, and other fibronectin molecules. These binding sites facilitate interaction that link these diverse molecules into a stable interconnected network. Binding activities of fibronectin molecule are enhanced by mechanical forces that pull on these fibrous proteins and unfold its component molecules. Unfolding of these modules expose binding sites that would otherwise have remained buried. Next point, binding sites for receptors on the cell surface. These binding sites hold the extracellular matrix in a stable attachment to the cell. The importance of a fibronectin containing substratum for cell attachment is illustrated in the chapter opening micrograph. The cultured endothelial cells shown in this photo are adapted a shape unlike any it would have in the body because it has spread itself over the available surface provided by a square coat of fibronectin. The importance of fibronectin and other extracellular proteins is particularly evident during embryonic development. Development is characterized by waves of cell migration during which each different cells follow different routes from the path of their embryo to another. Migrating cells are guided by proteins such as fibronectin that are contained within the molecular landscape through which they pass. For example, the cells of the nerve crest, which migrate out of the developing nerve system into virtually all parts of the embryo, transverse pathways rich in fibrils composed of interconnected fibronectin molecules. The importance of fibronectin in neural crest cell migration is readily revealed on a culture dish. A number of organs of the body, including the salivary gland, kidney, and lung, are formed by a process of branching in which the epithelial layer becomes divided by a series of clefts. Structure of fibronectin and its importance during embryonic development. A human fibronectin molecule consists of two similar but non-identical polypeptides joined by a pair of disulfide bonds located near the C-termini. Each polypeptide is composed of a linear series of distinct modules which are organized into several larger functional units illustrated by the colored cylinders in the figure. Each of these functional units contains one or more binding sites for either a specific components of the extracellular matrix or for the surface of cells. Some of these binding activities are indicated by the labels. The cell binding sites of the polypeptide that contains the sequence arginine glycine aspartic acid or RGD is indicated. As discussed later in this chapter, the sequence binds specifically to a particular class of integral plasma membrane proteins, integrins. RGD domain binds with integrins that are involved in cell attachment and signal transduction. Integrins are associated with cell attachment and signal transduction. The inset shows 2 of the 30 or so repeating Fn modules that comprise the polypeptide. The RGD sequence forms a loop of the polypeptide protruding from one module, a section through a young chick embryo that has been treated with fluorescent antibodies against fibronectin. Fibronectin is present as fibrils in the basement membrane, dark red sites that lie beneath the embryonic epithelium and provide a substratum over which cells migrate. When we look at the structure, we can see RGD loop, then the heparin and fibrin binding domain, collagen binding domain, and fibrin binding domain, along with the cell binding domain, then the heparin binding site, then the fibrin binding site, and COOH bounded to it, and other structures. So, when we take a cross section of chick embryo, young chick embryo that has been treated with fluorescent antibodies against fibronectin, fibronectin is present as fibrils in the basement membrane that lie beneath the embryonic epithelium and provide the substratum over which cells migrate. In this micrograph, neural crust cells are migrating out from a portion of the developing chick embryo system beyond the edge of the photo into onto a glass culture dish that contains stripes of fibronectin coated surface alternating with stripes of bare glass. The boundary of the fibronectin coated region is indicated by a white lines. It is evident that cells remain exclusively on the fibronectin coated surface. Cells that reach a glass substratum arrowhead tend to round up and lose their migratory capacities. The arrow indicates the direction of migration. The role of fibronectin in this formation of the embryonic salivary gland. The micrograph in D shows a mouse embryonic salivary gland that has been grown for 10 hours in culture. The gland is seen to be divided into separate buds by a series of clefts, open triangles. The gland shown in this E has been grown for the same period of time in the presence of an anti-fibronectin antibody, which has completely inhibited its cluster formation. Control can be seen and an anti-FN. So cells become, they doesn't even know where to migrate. So that condition is happening. The importance of fibronectin and other extracellular proteins is particularly evident during embryonic development. Development is characterized by waves of cell migration during which different cells follow different routes from one part of the embryo to another. So we will be learning about the importance of fibronectin and other extracellular proteins in particularly evident during the embryonic development. So what is embryonic development? What is happening during embryonic development? Development is characterized by waves of cell migration during which different cells follow different routes from one part of the embryo to another. Migrating cells are guided by proteins such as fibronectin, which are contained within the molecular landscape through which they pass. For example, the cells of the neural crest, which migrate out of the developing nerve system into virtually all parts of the embryo, transverse pathways rich in fibrils composed of interconnected fibronectin molecules. So, to guide the cells for the development of this embryonic body, we need proteins like fibronectin. To show them guidance where to move, where to migrate for that purpose. The importance of fibronectin in neural crust cell migration is readily revealed, and a number of organs of the body, including the salivary gland, kidney, and lung, are formed by a process of branching in which the epithelial layer become divided by a series of clefts. The importance of fibronectin in the formation of these clefts is illustrated by a figure which shows a salivary gland that has been incubated with antibodies that bind to fibronectin. Cleft formation and branching is entirely abolished as a result of the inactivation of fibronectin molecule. So, in case of development of salivary gland, there should be some clefts formed, which can also be induced by applying fibronectin. When we inhibit this fibronectin, there won't be no cleft formations, so that will be leading to an anti-fibronectin system. Along with, we will be giving a control system where we will be providing fibronectin, which shows cleft formation. But when we treat this salivary gland with anti-fibronectin substance, which shows no cleft formation. So that is all about fibronectin. And in this textbook of cell biology, there is a picture which we can see erythroid cells are there, lymphoid cells are there, primordial germ cells, and neural crust cells can also be seen. The role of cell migration during embryonic development. A summary of some of the cellular traffic occurring during mammalian development. The most extensive movements are conducted by the neural crust cells, which migrate out of the neural plate in the dorsal midline of the embryo and give rise to all the pigment cells of the skin. The sympathetic ganglia, adrenal medulla, and the cartilage of embryonic skull. The primordial germ cells migrate from the yolk sac to the site of gonad formation within the embryo. The progenitors of lymphoid cells are transported to the liver, bone marrow, and thymus along with the lymph nodes and spleen. These pathways, shown here, connect the original sites of cell with their destinations. They do not accurately depict an actual route of the cells.